Okay, so now let us get into the actual definition of biochemical or bioprocess engineering. So these two terms are sometimes synonymously used, biochemical and bioprocess engineering. But bioprocess, it tends to be more on the side of process. Process means we apply chemical engineering principles to the biological process. But biochemical, nevertheless, these are synonymously used. Okay, so biochemical engineering is defined as a use of microorganisms or part of them for the production of chemical or biological materials or the development of new processes. Okay, so like I said, we make use of microorganisms, and what do we mean by part of them? It means we produce some proteins or enzymes and we use it for different process. Say for example, we derive cellulases or some type of enzymes from microorganism, which we use it for some reactions for biotransformation reactions, or in the sensors, or for carrying out other types of hydrolysis reactions. Okay, so the product will be useful for further application. And here another important thing is for the development of new processes. So what it actually means, biochemical engineering, it not only implies that we use microorganism to produce some useful product, it also means that we use it for different application. So one simple application is environmental wastewater treatment application. So we consider all those big the vessels, or the plants that we are seeing, the tanks we are seeing as reactors, because microorganisms, they try to convert the large substrate into small substrate, which is what is happening in the fermentation process. But there the mechanism is more robust. So we see number of cells are grown, or they are naturally adapt to that condition and they grow, and then they're able to decontaminate the water. That means remove all the impurities present in the water, so that what you get out of the wastewater treatment plant is nothing but the treated water, so which can be used for like various other application depending upon the purity level. And then if you take this bioprocess engineering with chemical engineering as its core, when I say chemical engineering, like I said, we have been relying upon products from chemical reactions. So chemical industry means we will see large reactors where you have to handle all these chemicals at high temperature, which is the case in most of the reactions, and high pressure we have to give for reactions to occur. And largely we take the concepts of transport phenomena. Okay, so when we say with chemical engineering as its core, the part which as a user we can enable to the system. For example, when you grow the organisms in smaller scale in a kind of test tube or flask, the the agitation, we don't primarily concentrate on agitation or the aeration as a factor. Okay, so but because we provide try to give best conditions in smaller scale, but when the same organism is grown in a larger reactor, so what becomes more important than the growth of organism itself, the inherent capacity of organism itself, is how we are trying to facilitate all this, the chemicals, at what rate we are supplying chemicals and what rate we are transporting oxygen. All this, they depend on how effectively we are mixing the fermentation tanks. Okay, so these are the ideas, some ideas we always take from chemical engineering processes, because these are well established with respect to chemical engineer. So we take mass transfer, heat transfer, and momentum transfer ideas from chemical engineering, because these are three important things which we as a user have control over the entire fermentation process, because the organism, it has its kind of inherent or innate capacity which we don't have control over.
Sir, sorry to interrupt you. Sir, your mic is muted, sir.
Now am I audible?
Yes, sir, now you are clearly audible.
So when it started muted, hello?
Just now, sir. Just now, sir. You
Just
can
now.
carry
Okay,
on.
okay, okay.
Yes.
Dr. Bhushan. So biochemical engineering also embraces aspects of biochemistry, cell and molecular biology, bioorganic and bioinorganic chemistry. So this is a very important thing. We try to apply the chemical engineering principles, but one has to know about all the fundamental aspects, include, in fact, the advanced aspects of how the microorganism grows. So the areas such as biochemistry, cell and molecular biology, bioorganic and bioinorganic chemistry. Okay, so what is the role of bioprocess biochemical engineer? So when you walk into bioprocessing industry, like we see the process that we see, the production tank, and then we have what we call as a fermenter, and then the downstream section where we have a number of steps that are involved to purify the product. So what the so-called engineers who are working for a plant would be involved in? So it basically starts from R&D when organism is given to us, or a product is known to us, the type of product that we want. So the first step is to obtain the best biological catalyst, because there will be a number of microorganisms which can produce the same type of product. Okay, if you take ethanol, there are different types of yeast that can, most of the yeast can produce ethanol, but we have to select the best. And if it is a product through recombinant DNA technology, so you try to make a recombinant organism and get a protein produced, expressed from the the mammalian cell. We have to choose a right the expression system. Okay, which expression system would give us the good quantity titer of a product and the better activity of a product. So that is a very important role of the first role of foremost role of biochemical engineer. And then the next is to create the best possible environment. So here you should try to connect with the ideas that I like talked about in the previous slide. Okay, so what we as a user, as a bioprocess or biochemical engineer, has a control over the entire fermentation process. Okay, so we try to manipulate the variables, for example pH, temperature, agitation conditions, all these things. So you can see this particular image, so which tells what are the parameters, environmental parameters that biochemical engineers are interested in tuning up to make the production successful. And then another important thing is operating it in the most significant way. Okay, we have to always prefer the optimal conditions. And then the next is, once we identify the right conditions, we should know how to purify the product, because when you compare the microbial product, it will always have impurities as compared to chemical product. Chemical reactions, it won't have a number of impurities. So your your downstream section or purification section is straightforward. But for biological production, when a product is produced, it will always come with a number of other impurities. Okay, starting material contains a lot of impurities. Okay, so we have to come up with what would be the optimal steps or the condition that have to be put in sequence. For example, we try to extract the compound from other impurities, and then try to capture the target compound, and then we do intermediate purification, and then finally we go for polishing. Okay, so these are the steps which are like you will come across if you visit any industries. This is this is the the order with which the biological product is purified. First we try to reject as many impurities, as much impurities as possible, so what we call as extraction or clarification step. And then we try to capture the product of interest from the remnant impurities, and then we do a intermediate purification. All these we have to optimize the steps to produce a product in a most economical way. So which is another important job. We always want to produce a cost of production. Okay, because downstream processing, it consumes 60% of our investment, our operating cost in the whole fermentation or the biological production process. So all these are important roles of biochemical engineer. And if you ask, if you try to ask what the basic questions, for bioprocess development and design. So these are the answers for it. What changes, if you want to study, what changes can be expected to occur. So you need the understanding of microbiology, biochemistry, molecular biology, and genetics. This is very, very important. So like say if you go to any industries, you will see different, people from different background, so will be working, in a very coherent way, because without the understanding about microbiology, biochemistry, molecular biology, how to come up with, the right type of microorganism that is most, wanted for a production. So it is not possible. So we need the help of all these, different people to choose a right organism, and then to understand about, the inherent chemical reactions and the metabolic features of microorganism. And the next important question is, how fast will the reaction take place? This is where we take the help from chemical engineers. The chemical engineers or biotechnologists who know, like who have some understanding about the transport phenomena. So we talk about chemical reaction engineering, thermodynamics, transport phenomena, and biological reactions, and how can the system be operated and controlled for maximum yield. So that has to do with your process control and instrumentation. Nowadays we have all control sensors which can detect, and then the control system in place so that you maintain a proper temperature, pH, aeration conditions, agitation conditions, etc. And then how can the products be separated? We use separation techniques. Okay, so these are some advantages and disadvantages of biological process. The reactions are mild, so all temperature sensitive materials we are using, heat labile materials, even the microorganism, or like they are temperature intolerant. Okay, most of them you can grow up to 37 degrees centigrade. So whereas chemical reactions, they can be run at high temperature. And specificity, they have increased the specificity. That's why we are able to use them as, the kind of sensors as a catalyst for number of reactions. And effectiveness is one factor. And there is a possibility for using renewable sources. The one example is use of lignocellulosic substrate. And with the advent of recombinant DNA technologies, we are able to produce a large number of products, a number of proteins, that goes for say the proteins for cancer application or many other application. So largely they are based on the recombinant DNA technology. But there are also some disadvantages which like we are trying to address with the help of the modern technology. So the complex mixtures, as I told, they produce complex mixtures. So the downstream has to be very good. Then dilute aqueous environments, contamination is possible, but we have already kept this or completely kept this contamination problem at bay. So thanks to the kind of technology we have, the containment regulations, and variability with respect to batches, because we are dealing with the cell, and you cannot compare two cells. You cannot expect to produce or grow in the same way. Okay, so that is a complexity we see with respect to the growth of cells. That is the reason we see variability with respect to production. You compare two fermentation process, you won't be able to produce obtain the same titer. Titer means I'm referring to concentration of a product. So that is another important disadvantage. And now, we are able, like I said, we produce a large number of products, and these are some table shows list of products that we are that are in the market: ethanol, pectinase. These are the enzymes: glutamic acid, citric acid, some organic acids, insulin, recombinant insulin, which has been successful. And there are many Indian players in this particular market, insulin production. And then microbial protein, penicillin, all these antibiotics, you can see the applications where they are targeted for as fine chemicals. Say we use dextram as food stabilizer, citric acid for food products. Most of these products, citric acid is food products. Citric acid is used as a preservative, and of course, the use of antibiotics. And then these are products of recombinant therapeutics. That means we use recombinant DNA technology to get this macromolecules produced. Human insulin used to treat diabetes, erythropoietin for anemia, hepatitis B vaccine for, and these are, like I said, recombinant surface antigen based. So that is used to that access the the immunostimulator, gain immunity with the use of this hepatitis B vaccine for against hepatitis B. and many other products, interleukin 2, 11, which are used to treat cancers, GCSF, granulocyte colony stimulating factor for chemotherapy. All these are targeted for the cancer treatment and prevention. Yeah, a few other products of recombinant therapeutic route, interferon, streptokinase, which is used for acute myocardial infarction, tissue plasminogen, again it is used for acute myocardial infarction, and then follicle stimulating hormone. So there are a large number of products already in the market, and many more are being discovered, and their efficacies are being worked out, and then companies are looking for the right time to launch these products through recombinant origin. Okay, I think this particular thing Professor Satyanarayana has already talked about, but I'll give share with you few interesting incidents, the points uh, from this case study. So penicillin, if you go through the production process, it is so you can see the fermentation tanks that are meant to grow this, the penicillin notatum or penicillin chrysogenum. So it takes inoculum from a seed fermenter, and then we try to grow these cells and then give sufficient time with a feed of nutrients to it, and then it's a batch process. We have different types of processes: batch, fed batch, and continuous process. But it is largely done in a batch process to have a better economy of the production. And once a product is produced, that means penicillin, it will have a number of other contaminants. We call them as impurities, not contaminants, impurities. So we have to get rid of impurities first, like I showed in the previous or two slides before. So get rid of impurities. Firstly, we remove the cells using the filter. Cells is a major contaminant or the impurity in the product, and then we get the supernatant, and then we use it for further purification. For example, evaporator, crystallizers, and then what you get as a final product is the procaine penicillin, so which can be further derivatized to improve the efficacy of the molecule. So this is, like I told you, so the process development and then the development of design of the equipment, design of a process, they are largely they become the role of bioprocess engineer. Okay, so let me give you some interesting the takeaways when we look into the discovery of penicillin. As you know that Alexander Fleming uh, discovered this penicillin producing microorganism. So it all happened one day. He noticed that the plate in which he was growing some pathogens so was contaminated with a kind of a fungus. Okay, so he found something weird. This fungus didn't allow the growth of the contaminant microorganism which you are seeing here around the vicinity of it. Okay, around its surrounding you cannot see the growth of this microorganism. So this is a fungus I'm talking about, and it is forming a kind of clear zone. Okay, so at that time it was something new. Okay, so he found why it happens. Then he realized that this organism is able to produce a kind of antibiotic which has diffused out, and it doesn't allow the growth of the pathogens over it. So he found that it must be antibacterial. Okay, so that was a kind of thing. This is what you are seeing on the right is a Fleming's own photograph, and this is a molecule currently we are seeing as penicillin, but now we have a number of derivatives of penicillin with increased efficacies. And one important thing to note about antibiotics is as we see organisms becoming multi-drug resistant or antibiotic resistant, and at the same time we are seeing like we don't see the promising leads, antibiotic leads that can be broad spectrum even to this MR, the so-called multi-drug resistant the bacterium. So that is one for the past 10-20 years or so we haven't come up with a new the drug molecule, a potent candidate that can work against different types of microorganisms. So these are the two scientists who took up this work. So when he discovered penicillin, it was 1928, and then the two young guys, Florey and Chain, so they from Oxford University, they took it forward. They approached a number of companies, and then say the penicillin production started. Okay, so they initially started doing in batches like this. You can see tray type of fermenters, which have been completely replaced with the bioreactor, and then you see the growth of such culture in the bottles. Okay, that was a kind of understanding about the fermentation process long back that time. And then this was the first pilot scale penicillin production plant established at NRRL. You can see the use of milk cans as a nutrient tank to feed the substrate requirement to charge these fermenters. And then this is a first commercial plant, like established by Pfizer in 1944. Okay, so what were the issues that the companies were facing? So the yield was very low. The titer was very low, as low as 1 milligram per liter. It is not at all good. And there was poor understanding about bioproduction process, poor understanding about downstream processing, and then contamination was a major issue. And another important issue is the oxygen transfer, because we didn't have much idea about how the transport phenomena. That means how we can successfully transport oxygen to meet the demand of the cells for its growth and internal product formation. So all these issues, were undertaken. The challenges were addressed by bioprocess engineers, taking cues from chemical engineering processes. These are the contributions made by the production engineers. A new production medium based on corn steep liquor and lactose, which turned out to be very effective for production of penicillin. The anti-foaming agent, the role of anti-foaming agent was, its importance was the realized use of improved agitator designs and aerator designs, and to enable good, oxygen transfer, improved oxygen transfer, like systems were used, improved heat transfer was accomplished, and use of phenyl lactic acid as a precursor to promote the entire, the penicillin production process, and other, like aspects such as improved purification, use of mutant strains. I think these are largely, I think, addressed by Professor Satyanarayana. Okay, so these are the contributions, and finally, what happened? What was the outcome? Because penicillin production plant was established in 1945. Now we are able to achieve a titer as say 50 gram per liter, or even more than that. Okay, so this all could have been possible only these issues were sorted out by the so-called the bioproduction or biochemical engineers. So it was as low as 0.01, that means 1 milligram per liter. Now it is 50 gram per liter. That means 50,000 fold improvement in production with all these modifications made by the engineers. Now, yes, you can say it is the role of bioprocess engineer. They said like it requires help from many different streams: microbiology, molecular biology, biochemistry, recombinant. So only they can, with the help of them and understanding of processes happening inside the reactor, we can successfully design and develop a bioproduction process. Okay, so can I take a few more minutes?
Sure, sir. Sir.
Yeah, thank you. Okay, so next important concept which I would want to like say take for say another 1 or 2 minutes is see the mathematical models. As I said, I've shown you a slide where I talked about the ease with which you can grow the different types of cells in the fermenter, that enables bioprocess engineers to apply suitable mathematical models, because we rely upon these models to predict how the microorganisms can grow, and when it can start produce the product, what is the endurance of a process. So all these important parameters we can study, which are very important for large scale industrial production. Okay, so to understand mathematical models, see at what level we are looking at the microorganism. Okay, because we can treat the entire microorganism as one single kind of parameter. Okay, the compounded parameter called concentration. I don't go down to the individual cell, because you cannot see how 2 cells are growing or 3 cells are growing and compare the efficiency of their production capabilities. So rather, I try to take the entire population. I just sample out a medium, and then try to investigate the optical density of microorganism, or I go for dry cell way to get the actual titer of the microorganism or the product. Okay, so that is the one level of looking into the microbial growth. So we see the at the macroscopic scale taking one single parameter to predict the growth of microorganisms. And the other way is we try to go down at the cellular level and much down, that means intracellular level, how the different compartments work. So this information that I told, they largely help us in studying the cellular growth under different categories. So this is the most simplest case. So let me try to use this. So what is unstructured and unsegregated? The most idealistic case, that means we try to study the microorganism as one single microbial growth as using with the help of one single parameter. So where the cell population is treated as one and it as one component solute. But the most you see in the opposite, the most the kind of non-ideal situation, or what is actually happening is multi-component description of cell-to-cell heterogeneity. What it actually means, if you go down to the cell, you can see all cell machineries as separate compartments. How individual machinery, for example, the ribosome, if you try to compare the rate at which proteins are synthesized, and then you try to compare that with respect to another compartment, the rate at which nucleic acid is synthesized, we are going down to the cellular level to capture some essential information. But this is the most easiest thing, because we try to we can easily study this microbial growth in terms of cell concentration, but we have to apply all our understanding and then modern tools to study the heterogeneity in the system. So we always try to go from here to here, but there are two routes. So I'm not getting into this because of lack of time. You can also go through this route from here and then through this route to achieve this state of unsegregated and unstructured. Okay, so this is where we try to apply all these mathematical models. A simple mathematical model based on the unstructured and unsegregated is the modern model. Mu is equal to mu max into s by ks plus s. So I'm not getting into all this because of lack of time, but just to give a basic insight about how where we why we use mathematical models and what level we try to apply these mathematical models to predict some important parameters of the fermentation process. And mostly as bioprocess or biochemical engineer, we don't try to see much into what is happening inside the reactor. Okay, because once we feed the nutrients we try to optimize some parameters. I told the transport phenomena, and then we don't want to see what is happening, but rather we are interested in knowing about what goes into the system and what comes out of the system, to give a kind of they capture essential insight about the stoichiometry, kinetics of growth, and then the product formation. So all these are important aspect of biochemical engineering. Okay, so I am skipping this particular thing due to lack of time. So this is how this is a general route that is followed in bioprocess. We start with lab scale, move up to the lab scale flask, and then to lab scale reactors, and then to pilot scale, and then to industrial scale. This is a sequence with which all R&D labs, they try to establish, design a process or come up with the optimal conditions to establish bioprocessing platform to produce a certain product. So at different points we try to optimize, study some parameters, so that enables to achieve the industrial scale production economically viable. So yeah, these are the bioproduction environment, different safety levels that are adopted, and you can see how this biosafety level 1, 2, 3 are categorized. Basically, what type of differences you see in this. This is a simple scale. As you see the risk of organism increases, we add more and more safety equipments. So you can see the use of laminar chamber, double door entry systems, and then if it is a BSL level 3, biosafety level 3, the stringency with which the production unit is operated is very high, airtight. You can see the user is because he doesn't want himself exposed to the the kind of hazardous environment, biological environment. And this is BSL level 4, which all the recombinant vaccine companies they have to strictly adopt to this BSL level 4, which are the norms set by FDA. For any product that goes for therapeutic application, we have to adopt this BSL level 4. This is how the entire setup would look like. A typical setup for a BSL level 4 lab, it has 4 levels of operation units, and this is the actual lab. what you see here in the middle. At all other levels, platforms, they enable to maintain, the proper environment, the containment environment. So that is the norm of, bioproduction. This is the unit of inhalable insulin, the production plant. This is how it will look like. This is a fermenter, and you can see, large number of, this thing, which would be confusing to, like as the first time you are seeing this, but more you get into studying the process, you will understand what these are meant for. An inhalable insulin, fermenter, how it would look like from the top view, and then this is how the purification units would look like. And then even the formulation section has to be completely sterile. There is no human intervention at all. You can see the operator is inside the room. He is trying to control all this process, by having a good control system. And I think this again is a topic which Professor Satyanarayan has covered, like Indian biotechnology market. But just let me give few important points. So we have about 2,700 startup and 2,500 companies, and our share is 3%. The global share. And now we are, as per 2019, we are a 63 billion market and 12th among the world, third in Asia, second highest in US FDA patients. And we are the largest producer of recombinant hepatitis vaccine. But the vision is to achieve, to make this, the industry 150 billion. These are some examples of companies that are involved in biopharmaceutical production of, Serum Institute, which has tied up with, the Oxford to make this Covishield. Biocon, as you know, it's a very famous company producing large number of, different recombinant therapeutic proteins. And this is how Indian biotechnology market has been growing. It is slated to reach 1 or 2 billion. Sorry, it is billion. It showed us million. And then these are, the 4 sectors which bioproducts, biotechnology is categorized into. First is biopharma, where we are dealing with proteins, drugs, antibodies that are used for diagnostics and therapeutics. Bioindustrial sector, we make, enzymes, yeast, taste-based products. Bioservices, they actually augment both these sectors. They enable, the platforms for production of a compound. So research-based contract manufacturing contracts, clinical research contracts. Bioagri sector is related to biofertilizer, genetically modified seeds. This is important, like kind of the booming sector, genetically modified seeds. Bio IT, which augments, all these sectors, which tries to offer services to all these sectors, 4 sectors which are on the above. Okay, so with this I conclude my talk, and I'd be happy to take, if you have any questions, you can ask me. So thank you all.
Dear participants, can raise your question in chat box.
Thank you, Doctor. It's a wonderful section, sir. It's a very clear idea. You give that very, very clearly from the beginning to the current section of biotechnology. Thank you, Doctor. Thank you very much.
Thank you, Dr. Purushothaman, and congrats to the TANBio team, for having arranged this wonderful conference. And I think most of the participants are, they will start benefiting. So whatever I have covered so far, these are basics, and the understanding about the bioprocess is very essential if you want to get a job in bioproduction environment. So you need to have some basic ideas.
Thank you very much.
Thank you.
Anyone like to ask questions to Dr. Vivek Rangarajan? Anyone like to ask question? So Shailaja, you can ask your question now.
Hello, sir. Currently, you are talking about bioprocessing and mechanisms. The bio IT is emerging, right? Hello?
Yeah,
Can you hear?
These are already there. So only thing is, students are not allowed, because inside the premise they maintain contaminant high sterile environment. So suppose if you want to get internship or doing a project, industry setup, won't. That's why, the startups, like CanBio, they are trying to support, this kind of gaining knowledge on this kind of thing. So many more products are coming to market.
Okay, thank you, Shailaja. Some others.
Sir, I'm Kumar. I would like to ask a question.
Yes,
Yeah,
sir.
definitely you can shoot your question, sir.
Sir, biodegradable methods of biotechnology methods for kitchen waste degradation, sir.
Yes, sir.
Waste methods, please explain. Because it has to be taken in a bigger way. So, what do you suggest? Various ways.
Yes, sir. Like we are also using the waste from some food processing industries. But if I'm correct, like you are talking about the a kind of biopolymer, keratin, or I don't know the basic constituent of the waste. If I'm correct, it's keratin. They are keratin-based wastes. So I think we have some fungus that are capable of producing this keratinase. So that have been used. I think a few of my friends, they are also working on this particular field. So use of chicken waste and before the slaughter, you remove all the feathers and then it produces a lot of waste. And then we can think of making some interesting compounds. So if not, like we can biodegrade them. So to produce, even if it doesn't turn out, you are not able to get a product, but you can degrade them. But these are happening, I would say, not largely these works are taken up by big industries, but small scale units are already trying to address these issues.
Sir, kitchen waste, sir. Sir, kitchen waste composting method.
Oh, kitchen waste, sir.
Yes.
Oh, sorry. Yeah, kitchen waste.
Do you have any compounds developed some effective ways to compost the things?
Yes, sir. So that is a kind of well-established process. So when we deal with kitchen waste, like we use, the different types of environmental reactors. Say, for example, the best is to use, we call it as the packed reactor. So packed reactor or upward aerobic sludge blanket, anaerobic sludge blanket reactors. So these technologies are well established. And then we can also make use of this kitchen waste. If you want to produce useful product, first we have to convert them to the monomeric units. So which is driven by the enzymes. We use enzymes to convert them because they are of different categories. You will have cellulosic waste and then like starch waste. So all these have to be first digested to produce cellulose, and then we can use it for making product. For example, like as Professor Satyanarayana, like we can make use of those waste to produce some biopolymers. Because these organisms that produce biopolymers are much more robust. We can also grow them under high temperature conditions. I hope I try to address or answer your questions.
Yes, sir. Yes, sir. You are good, sir. Do you have such strains, sir, in your lab you developed?
We do have some strains, not specifically, to handle the kitchen waste, but we have some strains. But we have to figure out, like, do the studies how effective it can be on the kitchen waste.
Yes, sir. Okay, sir. Thank you very much, sir.
Thank you.
Thank you.
If anybody would like to ask questions. Okay, thank you very much, sir. Thank you very much for your presentations and your answers, which is most reliable to the society. Thank you very much, sir. Thank you for your support.
Thank you,
Thank you,
Dr. Purushottam. Bye.